All right, it's a great pleasure being here. Thank you for having me for this talk. So I'm going to go back to protein-DNA interactions, and what we may forget when we've seen all these beautiful structures of proteins processing our DNA, that in humans, the substrate is not DNA, it's chromatin, and that means that 75% of our DNA is wrapped into nucleosomes, forming structures like this. And to do structural biology on chromatin is quite challenging because it's big and it seems to be disordered as quite a bit. So the common consensus in the field seems to be that chromatin is disordered, or maybe, if you will, it's in liquid compartments. In half an hour, I hope to convince you that there's much more structure in it and that we need new tools to study that. And it all has to do with DNA sequence. So I'll have 3 parts. I hope I can do all 3 of them. My favorite tool is for spectroscopy and just taking chromatin fibers, so a couple of dozen of nucleosomes, and just tear these fibers apart. And we do that most often by magnetic tweezers, so having these beads attached to one side of the fiber and the other side to the glass, and we just pull on the bead. This is not to scale. The bead should be 5 times bigger, right? It is an electron microscopy image though. And then we just follow the bead and see what happens when you pull. And these are typical curves we get out when we pull on chromatin fibers. And when you pull very hard at 65 piconewtons, you get the typical overstretching behavior of DNA, and that's sort of the landmark of DNA for spectroscopy. Go a bit lower in force, we see small steps here, and if you zoom into this area, you can see that all these steps have sort of the same size, and that's when one nucleosome pops open, and then the next, and then the next, and you can just count them, right? If you would use optical tweezers, you can see the same thing, but you see this typical sawtooth pattern rather than this staircase type of behavior. But all steps are like 25 nanometers. Now this is all good and well. It tells you how many nucleosomes you have, but the interesting part is when it folds into higher order structures or not. That happens here at low forces, like below 10 piconewtons. And when you zoom in to that area in the experiment, you can see nice overstretching transitions as well, not unlike for melting the DNA. So here we're overstretching chromatin fibers. And what you can also see is that, well, we get different curves, but they have more or less the same shape. And we can fit models to these curves, and we get a fairly good fit for the single molecule data. And I want to talk you through the model that we have to fit these data, because the parameters that define these force extension curves, they really tell us about how the fiber is folded, how the nucleosomes interact, right? So this overstretching plateau we assign to unstacking of the nucleosomes. So the nucleosomes like to stack, and at 3 to 5 piconewtons they rupture, we tear them apart. And we know that because we could look at the stacking interaction. So this was already in one of the first crystal structures, and we can see that it's mediated by the H4 tails of these histone proteins, and we can make cysteine mutants, and within the same experiment we can covalently cross-link neighboring nucleosomes and then pull. And what happens then is basically nothing for the structure. You just get overlapping data, but there's not this overstretching transition. We make covalent links of stacked nucleosomes. So we are very confident that this overstretching transition can be assigned to unstacking of nucleosomes. There's more to it, and then it gets complex, because if you pull hard enough, anything breaks. So by looking long to these data, we've assigned 4 separable structures that we need to describe the data. And I'll talk through them from high force to low force. So at high force, we've totally unwrapped all the DNA from the nucleosomes, and basically when you're pulling, you cannot discriminate this from DNA. And if you cannot discriminate the force extension relation from DNA, we have a nice analytical solution for that, worm-like chain, and we can fit this nicely to this simple formula here. What is nice about it, if we have the relation between the extension and the force, we can just integrate it over the force, and then we also have the energy. Okay? For a singly wrapped nucleosome, the same applies, it's just a bit shorter. Same formula applies, same energy. For this one, we also needed to describe our data. It's again slightly shorter, but same thing happens. For the folded chromatin fiber, we don't know. And when physicists don't know what the energy landscape looks like, you just do a Taylor expansion. You assume it's quadratic, or you assume that it's a Hookean spring. So extension is linear with force, and then you can get the energy again. And when you have energies and extensions, then we can simply, by statistical physics, calculate the weighted average of the extension per nucleosome. And that's all what our model is about. Right, now let's forget about the formulas. I can just plot it. Here I've plotted these two formulas in one graph. So the energy is a function of extension. The red one would be for bare DNA. The orange one is shorter, but also lower in energy, because the DNA wraps around the histone core, and to break that costs energy. We can do the same trick here with slightly different energy. And then for the folded fiber, no matter what structure it is, the energy would increase parabolically, right? And there's here, this distance would be the energy that defines this equilibrium between stacked and unstacked and unwrapped nucleosomes. Same for delta G2 and delta G3. So by combining this, we can also calculate the average extension, and we can increase the force, and that basically tilts the energy landscape. And we can do the same thing, and we see the average extension increasing, and then we can see as this one gets lower energy, it becomes most prominent. So we have a transition here, and that can continue. So what I didn't tell you for these data is that we can control the locations of these nucleosomes on the DNA perfectly with one base pair accuracy by using a special sequence, the Williams 601 sequence, and just making arrays of these. Okay? And then you can use any sequence, and the cool thing about that is, and I don't have time to talk about it, but based on the sequence, you can just predict with similar statistical physics where the nucleosomes end up. And I can do that, but you can do it as well. Just go to the website, put your sequence in. There's a couple of parameters here, and then you can predict where these nucleosomes end. And this DNA sequence had a regular spacing of nucleosomes. So getting back to our model, I've shown you two data. They're of chromatin fibers with different distances between the nucleosomes. We call that linker DNA. And the red one has 20 base pairs between the nucleosomes, so ending up with a nucleosome repeat length of 167, because there's 147 base pairs wrapped around the nucleosome. The green one had longer linker length, 50 base pairs rather than 20. And that represents the average in human. 20 base pairs represents the average in yeast. And now, when we fit the model that I just explained, we get basically four parameters. The stiffness of the fiber, when it's still stacked. We get a unstacking energy, an unwrapping energy, delta G2, it's not plotted here. And that tells us more about the fiber. What does it tell us? Well, let's take this toy model. Suppose neighboring nucleosomes stack, and that's the case for the green curve here. It will have some stiffness. You can measure that by just pulling it. Right? So looking at the slope here. Now if you cut this stack of 6 nucleosomes in 2 and measure the stiffness of 3 of these nucleosomes, so a shorter chromatin fiber, the stiffness will increase. Basically you have less material, and it will double by stiffness, because you need twice as much force to get the same increase in length. Okay? Now if you would take 2 of these parts and put them in parallel, again you need twice as much force to get the same extension. So if we would go from a single stack to 2 stacks, 2 half stacks in parallel, parallel, the stiffness would increase by a factor of 4. Well, that's quite good. Stacking energy stays the same, and there's one ingredient that I did not mention, but you can already see it. Here we have a very sharp transition, phase transition. Here we have a gradual transition, and that's consistent with this interpretation that here each of these 5 stacking interactions can break, so that's degenerate, while here the bright red nucleosomes are much more fragile, than the dark red because they are stabilized. The stacking is stabilized by their neighbors. So this unstacks in a cooperative fashion, while this is unstacking in a degenerate fashion. Okay, so that's the basics. And you may say, well, this toy model, that's a very simple model for a very complex structure. And I fully agree, so we wanted a better structural model. And for that, we went to a package, Helix MC, that models DNA as just a stack of rigid base pairs, just domino stones. And each transition from one base pair to another is characterized by 6 parameters, and they can be curated from structures of DNA, and their variation can be curated from molecular dynamics. And by just stacking these in, say, with a Monte Carlo simulation, you can fully reproduce a worm-like chain. But we are not interested in the structure of DNA, we're interested in the structure of chromatin fibers, so we added a couple of ingredients. One, of course, is the histone wrapping. There's 14 contact points here that we fixed. The other one I already referred to is the stacking mediated by the H4 tails. We included them. And then we also need to include electrostatic repulsion between remote pieces of the DNA that are not in these parameters, right? And later I'll show you also that we can mutate these base pairs, and that will be interesting as well. So doing this, we can get a better structural idea of what's happening, and these are resulting Monte Carlo simulations, in which here I did not include any stacking, so I refer to a zero start. Here I only included stacking between neighbors, and here I included stacking between next neighbors. Okay. Now when you would plot the force as a function of this extension here, you get data that very much reproduce our experimental force extension curves. So this is really, I think, what's happening when we're pooling, and you could also see partial unwrapping and the unstacking taking place. What's the true value of these simulations is that we now get a much better idea about what the DNA between the nucleosomes does, right? And these are very short fragments, so using the assumption that it's a worm-like chain and an entropic polymer, that is not going to hold, right? And depending on the exact number of base pairs between the nucleosomes, you can see that there's a lot of stress building up in the nucleosomes, depending on whether you stack them in a one-start or a two-start fashion. And you can also see that when you add just 2 base pairs, that this stacking is really sort of effective. And only when you add 10 base pairs, the nucleosomes are parallel again, and they allow for stacking. I mean, here we forced it, so no matter what the energy. Here, with these short linkers, so 20 to 30 base pairs, there's lots of stress there. This is just not going to happen. This only works in our computer, right? We can do that for longer linker length as well. That's all nice and fine, but these are Monte Carlo simulations. But this motivated us to do our pooling experiments on a sequence of these fibers with well-defined linker lengths, right? And that's really revealing in the sense that when you start with 20 base pairs of linker DNA, you can see a fairly stiff fiber, 2 of these stacks in parallel, add 1 base pair, and it's inhibiting the stacking. Right? We don't see a lot of stiffness. There's residual stiffness. Anything has some resistance against stretching, but we interpret this as non-stacked nucleosomes, and only when you add 10 base pairs, we measure an increased stiffness again. Same for the stacking energy. Here we have large stacking energy. Here it's much lower, but remember the second conformation was not a full nucleosome, but it was a 1.2 times wrapped nucleosome. Okay, so there's already some unwrapping taking place, costs energy. The third transition, that's from this, bent, partially unwrapped nucleosome to a singly wrapped nucleosome. That's independent on the distance between the nucleosomes, because distance is too long, right? So we have a fairly good idea of what happens within those fibers, but this also has consequences for what happens in vivo, right? Because if these stacking interactions don't take place, the nucleosomes are free to stack with neighbors from other fibers, right? And when you do that, you introduce these phase-separated droplets. So here we mixed 172 base pair fibers, so these don't stack. Those were over there, right? And you can see that they readily form nicely globular shapes. If we go 5 base pairs less, where the nucleosomes stack in a two-star fashion, we can see very irregular interactions between the fibers, right? So these are not liquid droplets. This is not dynamic. And this is a way for cells to regulate whether the genome is in these liquid compartments or not. And it's coupled to a DNA sequence, which may make it functional for gene regulation. Let me skip these for the sake of time. Now, what really struck us, I thought, oh, this is all fine, nice model, we can work with that. And then we did the same experiment on an array of nucleosomes with DNA sequence, that came from a different lab, the DNA, and then we measured different values here, right? And then we looked at the sequence. Turns out that pulling on different sequences gives different fitting parameters. So, for example, here we have 2 chromatin fibers with different linker sequences, 20 base pairs, and you can see that the stiffness remains the same, but the stacking energy is very different for these 2 sequences. Right? This is a repeat, but then having 30 nucleosomes rather than 15, and that's our control, it's the same. And also for longer linker lengths, so 50 base pairs, we can see a significant difference in stacking energy depending on whether you have this sequence or that sequence. So when we want to understand higher order structure of chromatin, we also need to include the sequence of the linker DNA, and that makes for a big problem because sequence space is huge. We cannot measure them all. We did quite a few, and this is quite a lot of work. But then we can go back to our simulations again, and we can do mutations computationally. And in fact, we can evolve sequences such that stacking is optimized, or we can evolve to sequences that inhibit stacking. And then what you can see is, well, these are optimal for stacking, these are optimal for non-stacking. You can see a 10 base pair periodicity for the different dinucleotides here that either promote stacking or not. And that's all encoded in the genome, which makes it possible that genomes can evolve higher order structure, right? We're actually testing these sequences as we speak, so I hope that it actually matches our predictions here. Now, this is all fine, but could we get away from sequence dependence? Well, there's no DNA without sequence, but the best alternative we can do is have a periodic sequence with a period that does not match 10 base pairs. If that's the case, then moving the nucleosome around does not affect the bending, the sequence-dependent bending of the DNA, and you can put your nucleosome anywhere. And these sequences you can find in telomeres. Right? They have these 6 base pair repeats here. So we started to make chromatin fibers with telomeric DNA, right? With these 6 base pair sequences, and there should not be any positional preference. We did that together with Noort in Singapore. We made these arrays. They looked at it with EM. We can identify the nucleosomes. They're all there. You can look at the density. It improves if we put well-positioned nucleosomes at the very end, and then we have an array of nucleosomes on this telomeric sequence without any positional preferences. And we started pulling on them, and all the parameters were identical to making 601 arrays with the same number of nucleosomes. Well, that was surprising, because I just told you that, say, stacking depends on nucleosome positions depend on sequence. This did not affect the results, so both the unstacking and unwrapping energies and the stiffness. However, when I looked with cryoEM, we were in for a surprise. And here we saw that you can actually get very nice single stack columns of nucleosomes that are very well defined there. And you can see them in the original data. So nucleosomes like to stack, and if their positions don't affect it, they may even make super nucleosomes that are fully wrapped into chromatin. The stacking interaction is quite weak, and we also saw sort of gaped nucleosomes, so it can open up, it's quite dynamic. And because it's so weak in terms of pooling, we didn't see it. Now you may say, oh, that's nice, you can make that in the lab. No one has seen these telomeric chromatin fibers with these single stacks, and that's true. So we were looking for biological relevance, and then a player comes in that was already introduced this morning, TRF2. It's actually a telomeric protein that sort of organizes the telomeres. And when you slowly titrate in this TRF2, now you can see that the stiffness increases, the stacking energy increases, and even the unwrapping energy increases. And what's really important here, that this worked only for the telomeric chromatin. If you do the same thing for 601 arrays with the same density of nucleosomes, nothing changes. Right? So this suggests that there's really a biological relevance to this higher order structure of chromatin that we suggest that you find in telomeres. So it's all about linker DNA variations, and they regulate cis and trans stacking. But what does it look like in vivo? So for that, we ventured into another single molecule technique, namely methylation-seq, so nucleosome footprinting. Where do these nucleosomes end up as a function of sequence? We worked on that using DNA methylation by the HEA5 enzyme that methylates A nucleotides. And that's cool because A nucleotides are 1/4 of our genome, so we get a very high density of methylation, so very good resolution in its footprinting. And these methylated A's can be detected in PEG biosequencing. And actually, when you do PEG biosequencing, you're measuring both strands of the same molecule, so you get a 1 out of 50% potential density of methylation, and that gives us really high resolution. It doesn't only work in cartoons, it works in practice. So this is methylation data from yeast chromatin, and you can see that at some locations we get more than 80% methylation of the A's. Right? These are 100 molecules, and every black bar is a methylation. This is the sum of it. In terms of chromatin, it's more interesting to plot the size of the protected areas here. So when I do that and color code them, you can see many particles that have a size that are consistent with nucleosomes. And for each and every nucleosome, you can get the full distribution of nucleosomes along the array, and that will be sequence dependent. Now the reference here is MNase-seq, where you just digest all the linker DNA and do short read sequencing. When you do that, you get exactly the same positions, but that has, I think, more sequence bias and has arbitrary units. Here we get the absolute fraction of occupancy here, and we can see that at some locations, 90% of the molecules are wrapped into nucleosomes. Right, and you see nucleosome-free regions, that was well known, in front of genes, and you can even align all transcription start sites in yeast, and you can see that nucleosomes are not disordered at all. They are very well positioned at the beginning of a gene. There's a nucleosome-free region here, and if you sort them according to their size, you can see this nice curtain-shaped thing. Now, what we can do with these long reads is also look at correlations, because that's what these fibers are like. And we can see correlations up to 4 or 5 nucleosomes matching closely the 167 repeat, so 20 base pairs of linker DNA, not perfectly. And as we go for longer correlations, and they can last for a long time, the correlation length decreases. So there's definitely some order in these native fibers. What you can also look at is the footprint length, and here we were in for a surprise because most footprints were 123 base pairs rather than the documented 147, which means that some of the DNA is unwrapped most of the times. But what you can also see is that the next most common size is this 123 plus 147 base pairs. That suggests that we have 2 stacked nucleosomes here. Right? It's only 1 out of 100 nucleosomes that are paired, but they look like continuous stacks as we've seen in the telomeric chromatin. You can even see 3, 4, and maybe even 5 consecutive nucleosomes without any linker DNA in between. When you look at the linker length, it is also quantized in these 10 base pair steps, plus 5, but remember we also have 15 base pairs of unwrapping. So this actually corresponds to 30 base pairs of linker DNA, 20 base pairs of linker DNA, 10 base pairs of linker DNA. And we can see other particles, for example, the transcription complex as well with this long read footprinting. So stacking of nucleosomes mediate these fibers, and linker length controls which nucleosome stacks with what. It's sequence dependent, and we'd like to look at variations in vivo and at the dynamics, and that's something that this nucleosome mapping cannot do, right? And for that, we teamed up with German duo in Delft to work on a method to do single molecule FRET experiments, but then in a very high throughput. And we were inspired by using this next generation Illumina sequencing, and some people, some researchers have used this. After sequencing, they used the same sequencing slides to do affinity measurements. And with this sequencing slide, you make clusters of about 1,000 nucleosomes, and then you have a nice pattern of all kinds of different sequences. You know what it was, and you can look at what binds where. Problem is, this is very hard to translate to single molecule because there's 1,000 substrates there. There's also a lot of residual fluorescence there, and we're not going to look at single molecule binding, unbinding, unwrapping, wrapping of DNA. So we changed the workflow a little bit. We first did single molecule fluorescence measurements in time, and only then did the sequencing. Now, this seems like a very simple idea that anyone could have, and indeed we were not the only ones. And half a year before we were ready to publish, we realized that Sebastian Deindl was doing exactly the same thing. So we developed the habit of publishing back-to-back the same method, but cool outcomes. So the advantage of using these Illumina sequencing flow cells is for single molecule is that, well, there's no averaging, but we can have all the advantages of single molecule time traces, but then sequence resolved for a very large library. We did not test that on chromatin yet. I'd love to do that, but it's slightly more complicated. So we started with holiday junctions. They are sort of the prototype test for FRET experiments, and I'll speed up a little bit. We made a library of holiday junctions and varied the central 4 base pairs. Right? There's lots to talk about. You can also read it in the paper. We measure how many of these we have, and here we can see that GCs at this location are underrepresented, and we blame that to this ligase reaction. But let me get to, well, okay, you can see dynamics depending on sequence here, but not for all holiday junctions. Some of these holiday junctions did not show dynamics, and in fact, most of them are blue. Right? For that, we had to look closer into the structure. So these green ones, they are just nicely base paired holiday junctions. They allow for different conformations, different isoforms. Most of them are blue, they're mismatched. But when you look carefully, the static ones have a lot of A's and T's at these locations. They're just too fast. We can't measure them. The dynamic ones have lots of GCs, and the reason is that the intermediates, so when you go from one isoform to the next, these base pairs need to unstack. And that's again sequence dependent. And in fact, when you look at the rates, you can convert them to energy barriers, and when you relate these energy barriers that we fit to the ones that are assigned to these dinucleotides, you get a pretty good fit. You can even do better if you include, say, one more layer of base pairs there, then you get a very, very good fit. And you can even get the stacking energies of the base pairs from these experiments. I'd love to tell more about it, but it's very technical and time is over. We call this technique sparks, and we are on our way to apply this also to chromatin type of questions. Last but not least, I want to acknowledge all my collaborators, especially German Joe and his lab for the sparks. We did a lot of chromatin together with Lars Noorderschot and Daniela Rhodes, and the in vivo chromatin is together with Tineke Lindsna. Thank you for your attention.
Okay, so time for one quick question. Anyone?
How would you study the chromatin modeling? Using, for example, wouldn't it be like
we'd have to make a Fretz geometry where we can see changes in distance, for example, by labeling the nucleosomes with 2 Fret labels, or we can look at interactions between proteins binding to the nucleosome. So any single molecule experiment involving Fret can be translated to the sparks type of environment. No, the workflow is the same, it's just more of the same, so we... Oh yeah, we've also looked at binding interactions. We've looked at different conformations of the DNA, and we also have ideas of looking at protein structures. Yeah. Thanks.